nervous tissue a bit more in depth, and I want to go to the micrographs that I have here that you can see on your own. Before I get to showing you a micrograph, I want to show you where we're going to start. Oops. So this is actually a picture out of weird. Hold on a second. It's cutting off part of the screen, so I just want to try to get it all in there. There we go. So, anyways, this is actually a picture out of the next chapter. I just grabbed it because I want to show you what we're looking at. This is a drawing of, and this is a micrograph of, a cross-section of the spinal cord. And I'm going to show you a section that's essentially looking at this area of the spinal cord. In the middle of the spinal cord, there's a little hole called the central canal. So I'm going to use that as the sort of landmark so you can see where we're looking. In the spinal cord, the gray matter is this stuff that kind of looks like I don't know, butterfly outline or something like that. And the cells that are found down here in that gray matter, those are the cells that are responsible for making muscles contract. So these are actually the neurons that are the controlling individual motor units that we talked about last time. So that's what we're going to look at. Okay, I just wanted to orient you to what the whole thing looks like, because in the picture that I have here, it doesn't quite get the whole thing on the screen. Oh. And to be able to see this better, let me turn some lights on. Now, when I talk about gray matter and white matter, I'm really talking about fresh tissue, not stained tissue. But the way that gray matter and white matter stains, we can actually see very clearly where that is in the spinal cord. So first off, this is the hole right in the middle that I just pointed out to you, the thing called the central canal. And the line between gray matter and white matter is actually quite distinct, because just the background staining of the tissue is quite different. Where we see these very large dark spots down here, those are the actual neurons that we're talking about. These are the neurons that control motor units. Now, the way that neurons stain, this particular approach to staining neurons, they come out looking a little unexpected. The nucleus actually is going to stain fairly clearly. So there's not a whole lot of DNA in there picking up a lot of stain, except for this really dark spot that we see right there in the middle, or that we see up here or down there. And then the cytoplasm actually stains quite dark, especially if we compare that to the nucleus. That's the way that neurons stain, using a very specific sort of stain approach to it. Okay. So this is just a higher power image. This is using the 40x objective instead of 10x objective of part of what we just looked at. Okay, so here's the nucleus. It's kind of this little void in the middle of the cell with a dark spot in the middle, very middle of that. And then the cytoplasm stains in kind of a mottled pattern, has some texture to it. So let me go back to that 10x objective. The cell I was just pointing out to you is this one right here. Okay, and we can also see this cell right next to it at the higher power. We don't see a nucleus in this cell, although it's probably kind of where this little lighter streak is right here. But we don't see a very clear nucleus because we're not cutting through the center of the cell properly. This might be the edge of the cell. So this might just be a little pale spot in the middle where the nucleus might sort of be off to the side, but it's not a very clear example of a cell right there. When we see the nucleus in very clearly like this, or even a hint of it or a suggestion of it with less distinct edges over here, we're cutting through the middle of the cell. And this one, we just don't see much of the nucleus at all. So this is probably not anywhere near the middle of the cell. That dark spot right there in the middle of the nucleus is what's called the nucleolus. And the word nucleolus means little nucleus. It's just the ending on it in Greek, in Latin, is just, yeah, I guess that's Latin. It's just a diminutive, so it means little nucleus. What it's specifically for, this is the region in the DNA where genes for ribosomal RNA are found. So this means this is where ribosomes are constructed. What are ribosomes? Produce what? Energy. No, those are mitochondria. What are ribosomes?
Some proteins.
Say it again?
Something with proteins.
Something with proteins, yes. What with proteins?
They carry proteins, right?
They don't carry proteins.
They produce proteins.
They produce proteins. Yeah. Ribosomes are what translate genetic information directly into the structure of a protein. Okay? They're made out of RNA themselves, which is specifically called ribosomal RNA, as opposed to messenger RNA or transfer RNA, mRNA or tRNA, those sorts of things. So going back to the picture here, it stains very darkly right there because there's some very active genes, and there's also ribosomes being made right there. Ribosomes are made out of RNA. They're not made out of proteins. And so the genes are transcribed, and the RNA, once it's available, folds into the shape of a ribosome. Okay. Where do we normally find ribosomes, though?
I cannot seem to pronounce it, but aren't they on the endoplasmic reticulum?
You can pronounce that just fine. But yes, there's actually kind of two places we find them. Ones that are attached to the endoplasmic reticulum, and then ones that aren't. Okay, the ones that are attached to the endoplasmic reticulum make that endoplasmic reticulum rough. The ones that aren't attached are just free floating. But that's what's happening. Those are found out in the cytoplasm, whether they're attached or not attached to endoplasmic reticulum. That's where proteins are actually produced. So the ribosomes are made in the nucleolus, and then they're transported out to the cytoplasm where we see them here. And in fact, that's why the cytoplasm of the neuron stains the way that it does. The sort of little darker spots within the cytoplasm are where there are concentrations of ribosomes, probably associated with the rough endoplasmic reticulum. But it's not just a plain continuous color of the cytoplasm. There's some variation in the staining quality because there's some variation in where ribosomes are found. But the fact that ribosomes are very prevalent throughout the neuron tells you what about neurons? What are they doing primarily? They have a lot of ribosomes. What do? Yeah, they make a lot of proteins. Now, the proteins that neurons use are going to be cytoskeletal proteins, which give them their shape, like the branching shape of dendrites or that sort of thing. And they'll make enzymes to produce neurotransmitters. They'll make receptors that bind to neurotransmitters and those sorts of things. So there's a lot of things going on in neurons that involve proteins. And a lot of what we talk about next week is going to be based on membrane proteins like ion channels and those sorts of things. So there are a lot of proteins that need to be made all the time for neurons to function properly. And in the way that we've stained these neurons, we see the cytoplasm like this because there are so many ribosomes there. Now, we can see in this field of view other cells. So here are some smaller cells in the surrounding space, okay, that are obviously much smaller than these neurons we're talking about. Some of these might also be neurons. So this thing right here might be a neuron, but it's much smaller than what we're looking at, and its nucleus is definitely smaller than what we're looking at, probably because it's a smaller neuron. And what really determines the size of neurons is often how big and how extensive the whole thing is, including its dendrites and its axon. Now, for this one off to the side where we can't actually see its nucleus too clearly, we can see sticking off of it, there's a couple or few big things. So 2 sticking off there, and then a little hump right there and a little hump down here, suggesting where some other stuff might be coming off of it. Those are probably where the dendrites and maybe the axons attached to all those spots there. But this cell is going to be pretty large, taking up that space. This cell here is probably pretty small. If it has any dendrites of note, they're probably just within this little area right here. And then this cell, most likely, if it's a neuron, is connecting to some of its neighboring cells here. So if it has an axon, the axon's just going to be long enough to go out to one or maybe a couple of these neighboring neurons. These big cells here, their axons go quite far to their targets. So these are the cells that are controlling motor units. Their axons go out into the periphery along the length of nerves, and then they synapse at muscle cells, which is the neuromuscular junction we talked about last time. Yes?
If you had a different stain, could you like see the axons and dendrites, or would they show up if they were big enough? Or
Different stain. This is just a plain cell stain, which stains cytoplasm.
Yeah.
And there's something like cytoplasm in dendrites and axons, but it doesn't have the same composition as the cytoplasm in the cell body, so we don't see it.
Yeah.
But I'll show you some other pictures of neurons, not necessarily these, but I'll show you some different ways that we can visualize all of the structures of the cell. Yeah.
That last day, it was a Wednesday, it was May 13th, right?
Yeah.
Okay. I got
You know,
Switch
there's a
that
very
line
easy
up.
way for you to find that information out without asking the middle of me presenting stuff. On Blackboard, there's this schedule in the syllabus that tells you and everything else.
Oh, really?
Yeah.
Oh.
It's what I've been showing you up on the board all along. It was up on the board there, and I was pointing to it and saying 13th, 20th, and 22nd. And that sort of thing. It's in there. Trust me. Okay. Just as an aside, that's now been preserved for posterity because it's now recorded. It's on a YouTube channel. So everybody in the world will know that you just interrupted me to ask what day the thing's on.
I didn't know you were recording.
I always record. Or I usually record. So, yay.
Where's the camera?
There's no camera, it's just microphone. The
Oh.
video part is just what's on the screen. So,
Oh, well.
would you like me to state your name for the record? Anyway, so yeah, we'll see some other pictures and you'll see some variation. But these are neurons that are in charge of controlling the muscle fibers in motor units, as we talked about. One of these neurons might be responsible, say, for controlling the muscle fibers of my big toe. Okay. And so the axon has to go from this part of the spinal cord, which might be the lowest part of the spinal cord, is about at the lumbar region, the small of your back. So an axon could be leaving the the spinal cord at this level here and go all the way down my leg to the ankle and then turn to go to the end of my toe there to control muscle fibers in the toe. So that one cell has to be big enough to support that really long axon. And all of the proteins that are needed, the ion channels that we're going to talk about next week and that sort of thing, and the components of the synapse that are responsible for releasing the neurotransmitter and producing and recycling and all of that sort of thing, all of that has to be made in the cytoplasm. So that's being made here, and that's being sent down the length of the axon. And then the axon needs to have fibrous proteins giving it its shape and its strength. So all those proteins have to be made here. So they're really big cells, so they can support all of that. Now, the examples of neurons I'm going to show you, for the most part today, are going to be very big cells, so that we can kind of pick them out in micrographs pretty easily. But the fact that they're big is also going to correspond to that they have really long axons or really extensive dendrites. And we can see with this one off to the side that it has sizable dendrites. If this is a neuron right here, its axon might be shorter than this dendrite right here. Okay? So it's still quite a large neuron. Now, earlier I was showing you the little cartoons of unipolar, bipolar, and multipolar neurons. These are all multipolar neurons. You might not be able to necessarily see that. So this one I've been using that doesn't have a visible nucleus. We can see 1, 2 very obvious dendrites. And there's probably a dendrite or an axon coming off of here or off of here. So there's probably 3 or 4 things that we can see attached to this cell right here. And then the one that I've been using for the most part down here, we can see 1, 2, maybe 3, 4, maybe a 5th spot there, where there are things attached to it. Because there's not cytoplasm in the dendrites or the axons that we can visualize with a stain, we can't really see them all the time, but we can get a hint of where they probably are on the cell body. So this probably has several things sticking off of it, and this definitely has several things sticking off of it. But if you look at a cell like this, you could maybe say, okay, there's something sticking off of this end and maybe something sticking off of this end, but there's nothing obvious about the rest of the cell. That doesn't mean that this is bipolar. These are all multipolar cells. They have to be where we're looking. It's just a matter of what you see when you cut through the cell. Okay. So this is a kind of gruesome sort of suggestion of how to think about this, but let's go with it. If you want to tell how many limbs somebody has by looking at sections of that person, okay, you'd have to have a frontal section through the body where you can see both shoulders and both hips to know that there are 4 limbs attached there. If you cut through the body in a mid-sagittal plane, you're not going to see either arms or either legs. There's not going to be any limbs attached to it. So this might just be a section through the nucleus in such a way that we don't see a lot of things attached to it. Whereas this one, there's more things obviously attached to it. It's just a matter of where we cut through it in 2 dimensions to give us the image. But all of these are multipolar neurons. And in fact, all of the cells I'm going to show you are multipolar neurons, except for the next slide I'm going to show you. And those are unipolar neurons. We're not going to look at bipolar neurons. The only real bipolar neurons that are relevant to this course are found in the retina and the part of the nasal epithelium that processes smell, both of which are actually a topic that will be the very first thing we get to in A&P2. So bipolar neurons aren't even a topic for this semester. We're just going to be looking at things in the nervous system that are based on either unipolar neurons, which will just be what I show you in the next slide, or multipolar neurons, which are really everything else. So you are not going to be asked to look at a picture like this and decide if you're looking at multipolar or whatever types of neurons. If you ever were presented with that, the safest thing to do would be to call it a multipolar neuron, unless it's obviously not. And the only thing I'm going to show you that's definitely not is the next slide. And I wouldn't even say that's obviously not. multipolar, again, because it's just a matter of what you're looking at as you cut through this cell. Okay. So let me go to that other slide here. And the section I want to show you is of a structure called the dorsal root ganglion. So it's a ganglion, which means that it's a group of neurons, cell bodies in the periphery. The name of it, dorsal root, is getting at the fact that it's on the dorsal or posterior side of the spinal cord. Now, it's not in the spinal cord, it's just outside the spinal cord. And the root part of that is referring to the fact that it's the nerve root that's attaching to the spinal cord. Okay. So we can't see it terribly clearly in the picture right now, but we will see a lot of fibrous structures going through sort of in this area or up here. Well, we won't see this part. We'll be looking down here. And the fibers that we see are axons of the spinal nerve root that are attaching on the dorsal or posterior side. And these cell bodies are just clustered along that nerve root, and we call that the dorsal root ganglion. Okay. Now, in this micrograph, you can kind of see one really big cell there. Kind of stands out the way it's stained. But there's actually a whole bunch of cells right in this sort of patch right here, and there's some more down here that are this type of neuron. So here's that neuron I just pointed out to you, and using the 10x objective now. These cells are not going to really quite stain the way that we just saw. The staining approach here isn't quite the same, and they're not going to look quite identical. But on some of them, we can see a nucleus that's a little bit of a clear space with a nucleolus in the middle. This one doesn't quite look like that, but this one here, it actually does. And now we can see that, there's a nice big cell there. There are some smaller looking ones sort of in a stream right there. There's some here. There's a cluster there. There's some down here. They're just sort of all over the place. And then separating, say, this big cluster here and this cluster here, this is just a bunch of axons that are passing through the area. Now, these axons are the axons of these cells that we're looking at. Let me go to the high power of this. So this is one of those cells, and I don't really remember quite where I took this picture, but within that last frame. But this is one of those cells. We don't see a single thing coming off of this because it's just a matter of where you happen to cut through the cell to actually see it. So there might be an axon coming off the cell somewhere off of the plane of section that we're seeing here, and that axon goes up into the fibers right next to it and extends out in either direction. One end of a unipolar neuron, which is not labeled this way here, one end has dendrites, and then the axon goes from those dendrites all the way to the target of this axon. And the cell body is just off to the side. It has one connection out that gives rise to that axon. Now, unipolar neurons are really kind of found in only one sort of place, which are these ganglia. The dendrites of these cells are the nerve fibers that we see in the skin. They're going to be the sensory endings that are receiving information about touch and temperature and pain and pressure and those sorts of things in the skin, and into some other peripheral structures like joints and muscles when they stretch and that kind of thing. But the cell bodies are always in these ganglia. Okay. Now, they don't have to be in these dorsal root ganglia, but that's the most likely place that we'll see them just because there are so many of that particular type of ganglia. And those are the unipolar neurons that we see.
So unipolar are only found in the peripheral?
Right. Their cell bodies are always out in ganglia. And then the one end of the dendrite, and for some reason, as I'm talking about it, I always point to this side of the screen. So let's pretend this side of the axon is the one going out to the periphery. So it's got its endings in the skin or whatever. And then the other end of the axon goes into the central nervous system and synapses somewhere in the spinal cord of the brain. Now, again, these are really big neurons because they are going to be connected from very far distances. the dendrites of the cell that we're looking at right here might be in the skin of my big toe. So when I stub my toe somewhere, the pain I feel from that is being carried up this particular nerve. And then that fiber is going to go into the nervous system, central nervous system, into the spinal cord before it actually finally synapses. Now, there's actually two places, sort of generally speaking, where these fibers can synapse, which is the spinal cord or the brainstem. Okay? So if this is one coming from my big toe, the ganglion is going to be located right here next to the spinal cord, which in this model here, it's going to be at the base of one of these nerves right here. So it could be, the cell body might be down here, and then it synapses either right when it gets into the spinal cord, which actually pain fibers tend to do, or if it's in the end of my toe and it's not sensing the pain when I stub it, but I can like feel the texture of a carpet when I'm walking around in my bare feet or something like that. That kind of touch sensation will go up through the ganglion and then into the spinal cord, and it won't synapse until it gets all the way up to the brainstem, which would be right about here. So these really big cells can have axons that are, in somebody as tall as me, maybe almost 2 meters in length. So again, it needs to be a really big cell body to be able to support all of that fiber. There's not a whole lot of dendrites for this cell, just the ones that are in the end of, at the end of the axon in the skin. But it has a really, really long axon that goes to its target. Okay. So as I'm showing you this stuff in the group of pictures that I captured, there are two that say dorsal root ganglion, a 4x and a 10x sized magnification picture. And then the 40x is specifically called a unipolar cell. But that's where we find most unipolar cells. They're in this dorsal root ganglion. Okay. The other slides I was showing you a little bit ago were of the spinal cord. I showed you the 4x version, the 10x of the gray matter, and the 40x of the gray matter. Okay. So those are the three we're just looking at. When I get to talking about the glia, I'm going to show you the other spinal cord white matter pictures in a little bit. But for the gray matter, that's when we're looking at the neurons themselves. Now, the other picture I want to show you is of the cerebellum. Now, the cerebellum is a little portion of the brain. It's about 10% of the volume of your overall brain. The word cerebellum actually means little brain, little cerebrum. When we look at the brain, we usually think of it as being this big wrinkly thing here. This is what's called the cerebrum. This little piece here is the cerebellum, which would be attached, sort of hang off the back of the brain. It looks like a little version of the cerebrum. That's why it's called the cerebellum. Okay. And again, it's located right about here at the back of the head. The cerebrum and the cerebellum both have a lot of wrinkles to their surface. And so as we cut through the cerebellum, what we see in the slide here is sort of a section through one of those wrinkles. Okay? So here's the outer surface of the brain, and it kind of goes into the groove of the wrinkle here, and then another fold coming out on the other side here, and even some wrinkles deep inside, because it's just sort of all folded up on itself there. As we're looking at this tissue, all of this is gray matter. Okay, it's stained, but how it stains is, it makes it look a little different in regions. The kind of light part in here doesn't have a lot of neurons in it, but it's still gray matter because it's not white matter. It doesn't have the sort of fatty tissue making up white matter. And then the darker part down here, this is also gray matter, but it has a lot of cells all densely packed together. This region in here, where all these cells are densely packed together, is called the granular region or the granular layer because it looks like grains of sand. Okay? And then the lighter staining area out here has a name, which I'm not going to bother with because it's not so obvious from what we're looking at. I do want to point out, sort of in the center of the granular layer material, where it gets kind of lighter, this streak right here, this is actually white matter deep inside the cerebellum. Okay. So this might not look much different from this outer layer here, but knowing what's in that outer layer is, in the end, much different. Now, what we want to concentrate on are these kind of big spots that we see right at the edge between these two things. These are a special type of cell. So this is a 4X objective, and that's about all the detail we can see in it. I want to move to the 10X objective of this, where we see where these two regions come together. So that lighter staining part off to one side and the darker staining granular layer on the other side of the picture. And right in between these, we can see some really big neurons. Now we're looking at the neuron cell bodies. We're not seeing the rest of the neurons, but they are quite large compared to the other cells here. Now again, they're large because they have a lot attached to them. And what's attached to these cells are mostly dendrites. Now this picture here is actually a drawing of one of this kind of cell. They're called Purkinje cells. They're named after an anatomist. His name's Czechoslovakian. Not that Czechoslovakia exists anymore, but in the area that is the Czech Republic now. And it's actually would be pronounced a little bit differently in his native language, but most people pronounce that Purkinje. And the cell body is just this little part right here. So that's all we're seeing in the slide I just showed you. The cell is huge comparatively because it has a lot of dendrites sticking off of it, all kind of branching out into this huge tree out in this direction. The axon comes off this direction and has some branches that go to connect to some other things in there. But part of it's going to go down into that white matter that I showed you before. Okay. So if we're looking at this with this kind of stain, and this again is the 10X objective, this is the 40X objective, we can see the cell bodies here, but we can't see too much more. Now this one, we can see its cell body, its nucleus in the middle, and there's a little hint of something kind of going off this direction, which is where some of these dendrites are extending out of this area. Now, earlier I just pulled up a web page showing you a bunch of different pictures. I just searched for Purkinje cell with a Google image search, and I got all these things here. So the tissue that we're looking at right now is kind of like this. Every so often in the slides that we have, I can find a particular extension coming off of one or two of these cells that look like this, like the base of the dendrite. But most of them don't really have anything obviously attached to them like this. So this is kind of what we're looking at here. This is a drawing that somebody made of what they saw under the microscope. And they've highlighted what these cells look like. And they've sketched in sort of what they could tell of the beginning of the dendritic branching. But really these dendrites are going to extend all the way through this area here because they really look more like this. Okay? Now, this is a drawing made by an anatomist probably 100 years ago or more, staining these cells in a very special way that you can see all of the material there. Okay? Or here's a similar picture drawn by this actually rather famous anatomist who's passed away now. He was sort of at the height of his career around 1906 or something like that. But he used a process very similar to photography where he would fill the cells with a silver compound and then it would react the same way that you use chemicals to make film photographs develop out. And so he'd get a picture like this, and then he would trace it while looking at it through a microscope. So there are a lot of these kind of pictures made by that particular guy. And here are a few other sort of suggestions of the same sort of thing. Now, the colorful ones that we see like this is stained with a fluorescent marker. And this particular stain is just attracted to membrane lipids or to lipids. And so when it's injected into the cell, it basically fills the whole cell because it's sort of sticking to the membrane all over the place. And we can see the cell body very clearly. There's a hint of maybe the axon coming out there or maybe down here. And then as we see branches off of the dendrites, they get to be smaller and smaller, but we can see them extending quite a far ways out there. Okay. And there's a number of examples of this kind of cell. This one I kind of like. This one off to the side, this thing off to the side. This is a micropipette, which is actually injecting the fluorescent dye into this cell. And we can see where the axon's coming off on one side, and we can see all of the branching dendrites here. This one shows a whole row of these types of cells all kind of lined up with their dendritic branching patterns extending out into all of that range around it. Okay. So if you kind of think of this picture similar to this first one I showed you down here, especially the drawing, you can see how all of these cells are kind of lined up right at the edge of the granular layer, and the dendrites actually extend all the way out through the rest of the tissue. Okay. And there are a number of other examples of drawings based on different staining techniques, or some of them, like this one right here, is a computer reconstruction of a cell made by scanning the cell in basically 3 dimensions. So there's a lot of different representations of these types of cells. But really coming back to what we're thinking about here, it has a really big cell body, again, because it has a really extensive dendrite tree extending out into this area here. And if I go back to lower power, we can see the edge of the tissue right here. So between here and the cell body, this whole area here is filled up with dendrites. Okay, so all those dendrites need to be maintained by a really big cell body. Now, the axon coming off of this also does go quite a distance as far as neurons are concerned. This axon is going to go through the granular layer, and then down here where it starts to get a little faded out, that's the beginning of the white matter. And it'll continue through the white matter till it gets down deep inside the center of the cerebellum. That might only be a couple of centimeters in length, but compared to the size of the cell itself, centimeters are a pretty sizable difference. Okay. All of the cells in the granular layer, all these little dots here, for the most part are neurons also. But they're really, really small because, again, they have very few dendrites, and if they have an axon that goes very far, it's only going to go up into the tissue up here, something that can be measured in millimeters to micrometers of distance. Okay. So they're very, very small neurons there. Just to show you that there is some variation in size, but we usually pay attention to the big ones because they're the ones that really stick out. Okay. Now, in the book, this figure here where I was pointing out the Purkinje cell, there are a couple of other types of cells that are suggested in drawings. The first one here, or the third one here, the first one I want to show you, this is an example of where a bipolar cell is found. So down in here, there's a cell body. Its dendrites extend off this direction, and its axon goes up this way. Okay, and this is really something that has to do with stuff we'll talk about in A&P too. But I want to show you what they look like. Also, there are these cells up here, which have 4 things sticking off of them, which would make them multipolar. One of those comes down here and receives information from the axons that are down in this epithelium. And then some are reaching out side to side so they can interact with each other. Okay, so they have multiple structures coming off of them. They're not bipolar, they're definitely multipolar. And then their axon's going to extend up and travel away down here. Okay. The other one I want to show you, which is the first of the 3 in this picture, is another special kind of multipolar cell called a pyramidal cell. It's named that because its cell body is pyramid shaped or triangular in cross section, which in 3 dimensions would be either a pyramid or a cone, and somebody decided to call it a pyramid. And that's where it gets its name. The pointy top of the pyramid, the apex of the pyramid, gives rise to one dendrite that goes way up here. And then the base of the pyramid gives rise to a bunch more dendrites that extend around it. And then right out of the base of the pyramid is one axon extending out and branching to find a number of different targets. Okay. I don't have a picture of a pyramidal cell here just because we don't have a good slide of it in our slide set. So to show you pyramidal cells a little bit more, I'm just going to pull up a Google image search of some. Okay. And drawings like these actually kind of suggest the same shape I was pointing out to you. Here's a series of pyramidal cells found in a number of different places. There's some variation in their overall shape, but they're all basically the same, these pyramid-shaped cell bodies. And there are a number of different pictures kind of suggesting that. This micrograph here is the closest to what we have in our slide set, but I have not been able to find a very good slide in ours that even looks as good as this. Now, we can see the cell body here, and again, it's kind of triangular in shape. There's one axon extending out in one direction, and then dendrites coming off to the sides, and one big dendrite going from the pointy end or the apex of the pyramid. Okay. Usually, we would think of these as being oriented so they point up, kind of like a lot of these pictures up here. Usually, the apex is at the top of the picture. But this is just a random micrograph and happens to be looking at tissue that's oriented a little bit differently. But they show you a number of different spots there. Okay, so we don't have micrographs of that. I just wanted to show you a different type of multipolar cell. Okay, but we saw the Purkinje cell, and then the other type of cell, actually I should pull up a image search of that also. The first thing that we looked at is a motor neuron. Okay, the picture that we looked at in the textbook of the dendrites, cell body, and axon, where the synapse was, and the myelin sheath wrapped around it. That's sort of the typical example of a cell that we see, like a lot of these pictures here. Here's a couple, well, this is a micrograph, and then this is a drawing based on a micrograph of what these cells kind of look like in full shape. I was kind of hoping, oh, here we go. Subset of specifically cellular ones. Micrographs. So what we were looking at might look more like this or these here. This is a different type of staining procedure where we can see more of the dendrites and stuff sticking off of it. And this, we can definitely picture that as a multipolar neuron. But these are the kinds of neurons that are in the spinal cord that control muscle fibers. Okay. And there's a bunch of different ways to visualize them based on different staining techniques and that sort of thing. So this is pretty close to the micrograph I showed you earlier. A little darker looking stain, but it's still sort of the basic idea what we saw there. Okay. Okay, back to the book. That sort of wraps up looking at what neurons look like. What neurons do is really going to be the subject of what we talk about next week to go into the electrical activity. But we also want to look at these glial cells, which are the other player in nervous tissue. Glial cells come in a number of different types. There's 6 different types that we want to be aware of. 4 of them are found in the central nervous system, and 2 of them are found in the peripheral nervous system. In this table, I've lined them up so that we can see sort of the corresponding cells between central and peripheral tissues and what their basic function is. So we have astrocytes and satellite cells, which are both supporting cells. Astrocytes are star-shaped, which is what astro is referring to. Astrocytes are star-shaped cells Satellite cells, if you think of stars, satellites would be like moons or, artificial satellites. So they kind of both have a cosmic kind of sounding name to them. They're called satellite cells because they look like they're orbiting around a bigger cell. And I'll show you that in a micrograph in a little bit. They both basically do support. That's a very general term because what they do can be very general. They do things like maintain the concentration of ions in the extracellular fluid. And then they can also play a role in controlling what's going on in that tissue. The astrocyte is a good example of that. It's responsible for maintaining what's called the blood-brain barrier. Okay, and I mentioned earlier, one of the things that really sets the central nervous system apart from the peripheral nervous system is this blood-brain barrier. Now, it's not just in the brain, it's in the spinal cord also, but blood-spinal cord barrier just doesn't sound quite as catchy. What this represents is a privileged blood supply. Blood can't just get into the brain like it can get into most of our other tissues. Instead, there's almost like a wall or a barrier regulating what can leave the blood and get into the surrounding tissue. Basically, the only things that can get through the blood-brain barrier are very small particles like electrolytes or water molecules and essential molecules like glucose or amino acids or lipids. It's what we usually think of as nutrient molecules. Those are the kinds of things that can get past the barrier. And then other things can't because they're not allowed. And part of what keeps them from getting out are astrocytes. Astrocytes are sort of physically connected to the walls of blood vessels and will sort of not allow certain things to get out of the blood so that things that are potentially toxic to central nervous tissue aren't going to cause any problems. But in doing that, it's also so restrictive that there are a lot of things that we might normally think of as needing in the central nervous system that can't get there, like drugs. If you come up with a drug that can treat a neurological disorder, and you can do that in the laboratory under very artificial conditions, getting that drug into the brain really depends on being able to design something to get across the blood-brain barrier. And so taking advantage of the idea that glucose and amino acids can cross over very easily, drugs are usually designed to be carbohydrate or amino acid based, or lipids can cross over pretty easily because lipids just pass through cell membranes pretty easily. But larger molecules don't make it through very easily. A drug molecule like aspirin, which isn't necessarily that large, but it's not a specific kind of allowed molecule, it won't be able to cross through. Okay? Not that you'd need aspirin in your brain because you don't have pain receptors in your brain, but there is some selectivity is what gets through there. And that's largely what the astrocytes are responsible for. Satellite cells are kind of similar, although there's not really a blood-brain barrier kind of thing in the periphery, okay, because it's not the brain or spinal cord. There is some slight barrier that makes sure that really toxic molecules can't get in, but it's nowhere near the selectivity that we see at the blood-brain barrier. Okay. The next type, which in the central nervous system are called oligodendrocytes, and in the peripheral nervous system are called Schwann cells, they're responsible for providing the insulation around axons. And that insulation is what we call myelin. Okay, so myelination, another way of saying insulation. In the picture of the neuron that we saw earlier here, the cell off to the side, which is labeled oligodendrocyte, is specifically the cell that provides the myelination for axons in the central nervous system. Axons outside the central nervous system use a different type of glia called a Schwann cell. The way they work is very similar, but the precise way they do it is a little bit different. Oligodendrocytes can often provide multiple, maybe 3 or 4 different myelin sheaths around axons. Schwann cells, sort of the entire cell wraps around part of the axon, and we'll see that in a second. Also in the central nervous system are a couple of other types of glia. There are microglia and ependymal cells. Microglia are sort of the immune system cells of the brain. There's not a corresponding glia for the periphery because the periphery has an immune system. But the immune system's components can't get across the blood-brain barrier. So inside the brain and the spinal cord, microglia do what white blood cells do in the rest of your body, which is immune surveillance and phagocytosis. Ependymal cells are specifically, well, I shouldn't say only, one thing they do is they create cerebrospinal fluid. They also help to line the inner and outer, or well, mostly inner surfaces of the brain. Okay, and I'll show you that in a little bit. Now here's a picture, and let me make it a little larger, sort of depicting what glia in the central nervous system would kind of look like. So this row of cells here, these are ependymal cells. Kind of looks like an epithelium, but it's not. It's glial tissue. And they interact with some of the cells around them. This little fuzzy cell up here, this is called a microglial cell, which is kind of like a white blood cell. And then this big cell here, which somebody in the past said looks kind of like a star, is an astrocyte. And then over here we have a couple of examples of oligodendrocytes. The word oligodendrocyte means few, which is what oligo means, dendro, branches, which is what dendro means, and cyte, cell. So this is a cell with a few branches. So in these two examples that we see drawn in here, there's 4 branches coming off of each one. Okay, doesn't have to be 4 branches. There might be 3, there might be 2, there might be 5, depends on the oligo in question, but each branch is going to go out and wrap around one segment of an axon. So each of these myelin sheaths is made by part of an oligodendrocyte wrapping around an axon. This one comes from one cell, this one comes from a different cell, this one comes from the original cell, this one comes from the second cell. They're just, in this picture, alternating back and forth. It doesn't have to be quite that regular like that. There could be multiple cells that are all sort of contributing a little bit there, but those are oligodendrocytes. For the peripheral nervous system, this picture kind of shows us everything we need to sort of think about. Okay. Instead of looking at a region with a number of glial cells and at least a couple of neurons here, for the periphery, we're really just looking at one neuron, and this happens to be a unipolar cell, which has been cut open to kind of show you, here's the nucleus inside. Satellite cells are cells that are kind of wrapped around the cell body. And then Schwann cells provide myelin around the axon. Each of these myelin segments, or myelin sheaths, is one entire Schwann cell. Okay. Oligodendrocytes, they can sort of make up a few different myelin sheaths, maybe for more than one axon. But for Schwann cells, one Schwann cell wraps its entirety around the axon like we see here. So let me go back to the micrographs for a second and show you some of these glial cells. If we go to the spinal cord white matter at 40x, oh wait, no, I don't want to do that yet. We'll come back to that later. Let me just go into the spinal cord at 4x. In the white matter, the lighter staining area outside, which we can't see a whole lot of detail at this magnification, what we do see that are staining in there, some of what we see are little dots, which are nuclei of cells. The cells that are in the white matter are going to be glial cells. Exactly which type can't be definitively proven, but that's what we're going to see. Some of them in the white matter out here might be oligodendrocytes. Some of them might be astrocytes. Maybe a microglia, although microglia are really small and we can't really pick them out too well. Ependymal cells are very clearly seen in the spinal cord. They make this lining around the central canal. and this is at 4x. I have a 10x version somewhere in here. I thought it said, I thought I had one that said 10x of ependymal cells. But this is 10x, but it's showing us the neurons. Oh, there it is, spinal cord ependymal, 10x. So I was just showing you the same thing at 4x. Now at 10x, we can see a little bit more detail. And this thick purple line is just representing all of the nuclei of the ependymal cells in a row, kind of like an epithelium, but this isn't an epithelium. This is part of the nervous tissue. And while we're looking at this higher power, all of these dots out here in the white matter are cells, nuclei. They are probably oligodendrocytes or astrocytes. And it's most of what we're going to see in the white matter are those types of cells. Now, before I show you the last pictures in the micrographs here, I want to talk about myelin a little bit more. Whether it's in the central or peripheral nervous system, the way myelin forms is fairly similar. These pictures are specifically showing Schwann cells, which would mean it's in the periphery, but something similar is going to happen with oligodendrocytes in the central nervous system. The cell starts to wrap around the neuron, or around the axon. It'll start tucking in and wrapping around, making multiple layers of the cell membrane around the whole cell. Around the whole axon. In this drawing, the little oval off to the side, this is the nucleus. So for a Schwann cell, the myelin is thick layers and layers of cell membrane wrapped around the axon, and then the cytoplasm of the cell is sort of the outermost part where we can see its nucleus. Oligodendrocytes wouldn't quite look like this. The nucleus would actually be separate with a branch extending around to wrap, a branch extending out to wrap around the axon. Now, this figure here is an electron micrograph. Using electron micrograph, we can make much higher power images than we can with a light micrograph. With a light microscope, we can usually see a few hundred times magnification, maybe up to 1,000 times magnification with a really good microscope. But with an electron microscope, since we're switching from using light as a source of particles to electrons, we can get higher and higher resolution. Now, this is a picture from the University of Michigan Medical School, which has a fairly extensive microscopy website. And the magnification factor that we see here is almost 1.5 million times magnification. Okay. When we look at the micrograph at this magnification, we can see quite a bit of detail. And let me just enlarge a little bit. Now, electron micrographs are, by definition, monochrome or colorless. Color comes from photons, from light particles. And we're not using light particles here, we're using electrons. When we're capturing an image of an electron micrograph, we're just putting some photographic film on the opposite side of the specimen from the source of electrons. And when the electron hits the film, it exposes the film there. Now, it does this in a negative process. So everywhere that electrons hit the film, it's light in color. Okay. Anywhere that electrons are blocked from hitting, it's dark. So the dark areas are like shadows that are cast on the film. Just like if I put my hand up in front of this white part of the screen, the shadow behind it is where light from the projector is blocked, so it can't hit the screen up here. That's what's going on making a picture here. So anything that's dark is where electrons are blocked. Now, electrons are really, really small, especially compared to the atoms that are making up molecules in here. So they're going to pretty much pass through the tissue unimpeded unless we put something in there to block them. And in electron micrographs, what we block them with are heavy metal atoms. The two that we use primarily are osmium, which is associates with lipids, and lead, which associates with proteins. So knowing what we're looking at, or at least I know what I'm looking at, I can tell you what things are. These layers and layers here, which we can really see the distinct layers in this region, it gets a little fuzzy off to the side here, but those layers are cell membranes. So they're visible because the osmium's reacting with the lipids. And then in the center here where the actual axon is, we see some proteins and some lipids. Now, all of the little dots that we see in the cytoplasm of the axon, those are fibrous proteins that make up the structure of the cell. It's cytoskeleton. This thing right here is a mitochondrion. Okay, mitochondria have a very particular appearance under electron micrograph. So there's a mitochondrion here, there's a mitochondrion here. What we see right here, kind of pulling away from the dark, thick wall, is the cell membrane of this particular axon. Okay? It shouldn't pull away like that, but as this tissue has been treated for microscopy, we do see a little bit pulling off there. This big structure here that's sort of blank in the middle, this is probably a vesicle, which is moving along the length of the axon from the cell body out to probably the end of the axon, and it might be carrying some important nutrient molecules or something inside of it. Okay. So we can see these structures in here, knowing what we're looking for and knowing what kind of things are stained, we can pick them out pretty well. Okay. Now, this is a great kind of micrograph to show you just the layers and layers of layers of cell membrane that wrap around the axon, which is the myelin. Okay, it's very similar to, oops, where'd that picture go? This picture here. Okay, so just layers and layers of cell membranes wrapped around the axon is what we see in the micrograph down there. Now, having showed you that, I want to go back to the micrographs that I've captured to show you a couple more. First, I want to show you this. I should say the 3 micrographs I'm going to show you here are all stained the same way. They're stained with an osmium kind of stain, which means that it associates with lipids. These are light micrographs, not electron micrographs. But it still works the same way. The same way that I can put my hand up in front of the screen and make a shadow with my hand. Where the lipids are, osmium stains with it, and they make a shadow on the film that we're taking this picture of. Or I should say, not film. This is on this microscope up here, so the light passing through the microscope is blocked, and what gets projected up on the screen are dark spots where the lipids are. Now, this is a nerve out in the periphery. The nerve itself is just this thing up in the top corner that kind of looks like a slice of watermelon. And we'll look at that in higher power in a second. But what I want to point out to you is the rest of the tissue here, since we're out in the periphery, it's not just nervous tissue. This tissue here is actually a different type of tissue that you've looked at before. But the way you've looked at it before, it was stained differently. Here we're staining the lipids, and so we get this kind of pattern of all these dark spots all over the place. The way you've looked at it before was with a cell stain, which does not stain the lipids, and you would just see the cellular component of this tissue, the nuclei and cytoplasm of these cells. So what you've seen before would actually be kind of a negative image of what we have here. But based on how I've described it, we're staining here and comparing it to what you've seen before, what type of tissue would this have to be out here on the side? What type of tissue is going to have a lot of lipids in it and pick up a lot of this osmium stain? Well, lipids are what's staining. What type of tissue has a lot of lipid in it? Come on. Tissue with a lot of lipids.
What's it called? Adipose?
Yeah, adipose tissue. Okay, fat tissue. These dark spots here are the fat globules that are inside adipose sites. Okay, that's what we're seeing here. So I just want to take that opportunity just to show you what this tissue looks like and get you kind of thinking about what we're staining here. Okay. If we go to the higher power, this is 10x version of this. The nerve has been cut in half, kind of split open like a banana split. And then that half of the nerve has been sectioned. So we're looking at cross sections of half of a nerve here. If you look closely in this tissue, you can see some places where there's a dark line surrounding an empty space, which is very similar to what we just saw in the book here, this kind of thing. Okay. Now this is 1.5 million times magnification. This is a 10x objective projected through this system. So this might be 100 times magnification. I don't exactly know all the magnification factors involved in the digital camera here and the projector and all that sort of thing. But it's definitely a lot less magnification than the electron micrograph I was showing you. But we can still see something reminiscent of what was in that electron micrograph. Okay. All of these are axons cut in cross section. We can see the myelin wrapping around it. The center, when there's a visible center, is blank because the center of the axon isn't picking up any stain. Now, this isn't the kind of stain like in the electron micrograph. We're not staining proteins and lipids. We're only staining the lipids. But we can see some structures in here, which is obviously myelin wrapped around some other structure there. Okay. And they're scattered all throughout there. Now, I started to show you this spinal cord white matter high power image, and now I want to show you that. Based on what I just showed you, now we're looking at a cell stain, and the myelin does not pick up any stain. So where we see the light staining here, or I mean the unstained portion of this tissue, that's the myelin. The little faint dark thing in the middle, that's the axon, which has a little bit of protein in it and is picking up this cell stain. But this is essentially a reverse image of what we just looked at. This happens to be central nervous system tissue, and what we're looking at before was a peripheral nerve. But they both have myelination in the same way. Okay. So thick lipid surrounding the axon in the middle there. Okay. So that's just to point that out. Now I want to show you one last micrograph here. This again is stained with the osmium. This is a teased nerve, which means a nerve split open the same way as the watermelon shaped section I showed you a second ago. But instead of sectioning that split nerve, now the individual axons are teased apart so we can see them individually. But they're stained with that osmium stain so that the lipid in the myelin stains black. Where we see a little space like this, that's one of those nodes I was talking about, a node of Ranvier. So the myelin ends, there's a little gap, and then the myelin starts again in the next segment. Okay. What I like to show in this picture, what I like to make a point of, is that the distance from one node to the next is actually really, really long. Let me remind you, when we're looking at the neuron before in this picture, the myelin sheath looks like it's a little packet, and then there's a node, and a little distance, another one. This is not drawn to scale. Okay, it's very important that you understand that this is not what myelin really looks like. Okay, this is just for the sake of a textbook figure. This is what myelin really looks like. So going from this spot right here down to this spot right here, that's how far apart the nodes actually are. Okay. This thing right here, that's a little crushed part of the nerve. So the stain got messed up right there. But we can sort of see that there's an end and an end on either side of that gap. We can see where two ends kind of make space for a gap right there. That's definitely a node. This is definitely a node. This is definitely not a node because it doesn't look right. Okay. Now, another reason why I know that's not a node is because this definitely is, and nodes are not that close together. They've got to be that far apart. Okay. Over here where we have some more fibers, we can see some other things that might be nodes. That might be a node there. That's definitely a node right there. That's probably a node right there. Some of these things look like where the fibers got crushed in the process of teasing it apart. So we can't be entirely sure. And then a lot of these fibers kind of end up crossing over each other. But with this one fiber, we can follow it along from one node and find the next node right there. We can see this node here, and if we follow this fiber along, we go off the side of the picture before we find the next node. Okay, but these two are both within one frame together, so it's kind of nice to pick that out. I think it's important for you to see more realistically how the myelin sheaths are wrapped around these axons, because they're not as short as a textbook picture ever makes them out to look. And oh, I think I closed the search window I had earlier, but when I searched for motor neuron, or I think that's what I searched for earlier, there were more pictures kind of representing this sort of thing. Again, it's not drawn to scale. If this was drawn to scale, based on the thickness of that axon, the distance from one node to the next would go off the page. Okay. So to make it all fit on the page, it's necessarily going to be, they're going to be closer together like this. But anyways, so that's sort of showing you the different types of glia as we see them in these pictures here and here, and under the micrograph, in the micrographs like this. And I explained how myelin forms. Yeah, and that's the end of that part of the chapter. Okay. So that kind of wraps up talking about the nervous tissue stuff. Then we're going to get into looking at how nervous tissue functions, how these cells that we're talking about connect with each other so that electrical signals can be shared and we can actually carry out some of these sensory or motor functions that I mentioned earlier. And a lot of that has to do with the electrical activity or the nerve impulse, which we already defined or named as the action potential that came up when we were talking about the muscles. And then we also want to consider what's happening between neurons, which is really getting at the synapse. So we'll look at that structure in more detail beyond what we learned for the neuromuscular junction. Okay. So that material we're going to cover next week. It's entirely possible that that will take the entire time next week, and we might not finish up the rest of the laboratory material. So again, oh, by the way, Stephanie, that's what it looks like. You want to see where all the times are.
I've seen it before.
Oh, good. Well, you can look at it again. So the next week folder right now has the neuroanatomy lab quiz. Some of the questions there have to do with the pictures we were just looking at, but other questions have to do with the other lab material we're going to go on to do. It's possible we won't have a chance to complete that next week, or we might spend the whole time talking about electrical activity. If that's the case, then I will push the due date for the lab quiz back by a week, if I haven't already. Yeah, so right now it's set also for the 26th, which would be before the week 12 class. But if we don't get to talking about the rest of this material until the week 12 class, I'll push this back by another week for you to complete it. And it just might be that next week we won't really have time to get into that. And what we're going to do is we're going to look at models of the brain. I pulled this sort of half brain model out of the torso model here earlier, but we have other models of the brain. We'll talk about the structures that we can see on the surface of the brain, and a little bit about how that stuff kind of works together. So when we get a chance to, we'll start talking about that. But so that kind of wraps up what I wanted to talk about today. Of course, we're finishing up a little early, but the material in this chapter is a little bit dense. So today I just want to talk about the anatomy of the tissues. And then next week we'll get into looking at the electrical stuff. If I start talking about the electrical stuff today, I just have to start it over again next week because you just need to have it all kind of together there. So we'll go ahead and call it a day now. And we'll come back next Wednesday on the 22nd to continue this. So, and remember the 20th is a holiday. School will be closed. If you show up, they won't let you on campus. And anyways, so. Otherwise, have a nice extended